So I've talked a lot about LTP, and to be honest, I've only just touched the surface, right? Synaptic plasticity is a huge field. There are all sorts of different forms of synaptic plasticity. I'd be lying if I didn't tell you that, there are also NMDA receptor independent forms of LTP. Every single form of plasticity I've mentioned here has been at an excitatory synapse, and there is strong evidence that inhibitory synapses can also be plastic. But there's even more forms, right? One that I've got to touch on is intrinsic plasticity. So here we see what is known as homeostatic plasticity. So they get some cells growing in cell culture, and they inject current into them, and they get this white triangle line here, right? So this is the input-output curve, the IF curve of the cells on average. Then what they do is they apply a drug that blocks all excitatory input to the cell for 2 days. And when they come back, they need to apply less current to get the same cell to fire. So here's the same amount of current, and the cell now is firing like crazy. So this is called homeostatic plasticity because it seems like the cell is trying to match the rate at which it fires action potentials, right? So before they put in the drug that blocked all the AMPA receptors, let's say the cell was firing, you know, once a minute, and then when they blocked all the AMPA receptors, the cell was firing 0 times a minute, and the cell thought, oh, this is weird, this is wrong, something's gone wrong here, I need to change something. And what does it do? Well, actually what it does is increase the expression of voltage-gated sodium channels. So here's the sodium current, here is the voltage dependence of activation, and you can see that the activity-deprived cells, that is the ones that have had all of their input removed, have a current that's about 50% larger. But the voltage dependence is exactly the same, right? So it's nothing to do with the voltage dependence. It's just that there's more voltage-gated sodium channels have been inserted. Uh, you can even see um, intrinsic plasticity with more subtle experiments or more subtle manipulations and more rapidly, right? So that took 2 days of blockage of every single AMPA receptor. So this study, we'll go through it a little bit more slowly, right? So they're stimulating these presynaptic inputs. They've got a postsynaptic cell that they've patched, and they do theta burst pairing basically between when the cell is firing action potentials and when the input comes in, and they do that just right. And what you see is here is the EBSB getting larger and larger and larger. Boy, it's getting large, nearly 3 times the size of when it started. Again, if they put on AP5 and MK801, both of these are NMDA receptor antagonists, nothing changes, all groovy. But when they inject current to the cell, what they see is that the voltage response is smaller after this theta burst bearing. So you can see the change in voltage is less post the theta burst protocol or theta burst pairing, right? So what does that mean? So that means V equals I R equals V over I. So V has gone down, I has stayed the same, so R has gone down. So the resistance of the membrane has gone down, or the conductance of the membrane has increased. And here we see the input resistance before and after the theta burst pairing. You can see it's dropped from about 60 mega ohms, there's bad units there, down to sort of 40 mega ohms, right? While when they put on the AP5, that doesn't happen. And here you can actually see the input resistance dropping over time. Um, so what is happening here? So the important thing to notice is they've done a classical synaptic plasticity type experiment. They've induced the synaptic plasticity. It's NMDA receptor dependent, but at the same time, it appears that the ion channels, the non-synaptic ion channels in the cell are changing. Here they are applying the doing an IF curve, an input output curve. Here's the injection, here's the spike, and they see that over time this curve shifts to the right. So the cell is getting less excitable, even while the synapses are getting stronger. And what they show is here it is prior to the theta burst pairing. Here it is after it, so you can see that they used to get a big train of action potentials, and now they only get one or two. But when they apply this drug ZD7288, which blocks an ion channel called IH, Then they go back to baseline. So what they show in the rest of this paper is that this form of theta burst pairing, while inducing synaptic plasticity, also changes the behavior of the cell by inserting these IH channels.